This is an explanation of a single mutation in the prM protein of Zika virus contributing to fetal microcephaly. The Zika virus outbreak began in Uganda in 1947 and continued on to Southeast Asia, then Micronesia in 2007, and Central and South America in 2015. Due to its increased prevalence, in 2016, the World Health Organization declared the Zika virus a global epidemic. There are two lineages or strains of the Zika virus, the African and Asian strand. The Asian strand of Zika virus, CAM 2010, is the version that mutated to become more virulent and deadly to the fetal brain. This mutated form of Zika causes microcephaly, an occurrence during which a baby's head is underdeveloped during pregnancy. This can be present at birth or can develop in the first few years of life. Babies who survive often have intellectual disabilities and impaired motor functions. In one of the earlier correlations, a pregnant European woman working in Brazil developed Zika-like symptoms. After returning to Europe, ultrasonographic scans showed that the baby's skull was severely underdeveloped, consistent with microcephaly. Although the subsequent study could not conclude with certainty that Zika virus was responsible for the microcephaly, this case, along with similar ones from across the world, was enough to arouse response within the World Health Organization and leading scientists around the world. The question then became, what is making these new strains of Zika virus more dangerous? A previous study investigated the effects of Zika strain on the embryonic mouse brain. The results showed a direct effect of Zika on neural progenitor cells, or NPC development, including cell death, which correlated Zika with the development of microcephaly. This correlation was further compared between fetal humans and neonatal mice. The ancestral Asian strain, CAM 2010, was compared to all three contemporary strains, MTQ 2015, VEN 2016, and SAM 2016 in neonatal mice. MTQ 2015, VEN 2016, and SAM 2016 were all lethal, and CAM 2010 was not lethal. In the above figure, the CAM 2010 Zika strand proved itself the most deadly among the mice sample, and the MTQ 2015 strand seemed to be the least effective. The SAM 2016 and the VEN 2016 strands were about equivalent in the effectiveness among mice. In the above figure, in order to determine virulence, 7 mutant viruses or outbreak strains were found: T106A, S109N, N130S, S193N, K709R, A982V, and N31443. T106A and S193N evolved differently than the rest, and S139 had the greatest neurovirulence in neonatal mice. In conclusion, the results of this study indicated that the ancestral Asian strain of CAM 2010 resulted in mild microcephaly, while more contemporary strains were much more lethal. The N139S mutant virus retained its neurovirulence. And that's the mutation in prM protein of Zika virus contributing to fetal microcephaly.